Welcome to the Organic Chemistry Podcast, Dr. Brian Lloyd's scribblecast of organic chemistry lectures and solutions to homework problems. In this scribblecast lecture, we're continuing with our study of carbohydrates and are going to look at biologically important derivatives of monosaccharides. Now, clearly, one biologically important derivative is the glycoside. The glycoside we've already covered. If you recall, it is that which occurs when you take a hemiacetal, such as alpha D-glucopyranose, and you react it with some kind of alcohol. Now, this alcohol could be another carbohydrate, or it could be something as simple as methanol in anhydrous HCl. If you do this reaction, you will produce OCH3. Now, in actual fact, although I'm drawing it alpha, just so I can name it, you would get a mixture of alpha and beta in this reaction. Enzymatic reactions can often retain stereochemistry. So if there was some enzymatic process producing a glycoside, you may get one specific anomer. Nevertheless, our alpha D-glucopyranose would become an alpha D-glucopyranoside, and our hemiacetal would have converted to a pyrano. I'll put a hyphen there so I can finish it. Side. And to indicate the methyl group up front, I'd put methyl as a separate word up front. Now, this is a glycoside, and glycosides are important, and the glycosidic linkage is important biologically because it is the linkage by which monosaccharides will polymerize into di, tri, and polysaccharides. And we will be looking at that a little later on. Another very important class of biologically important derivatives of monosaccharides are the sugar alcohols. Or what might be called the alditols. Now, if you take the straight chain form of D-glucose and you reduce it. In biological systems, this would be done by an enzyme, but in the laboratory, we can use H2 gas and platinum as a reducing agent. Or, alternatively, we could use sodium borohydride. Either reagent will take the aldehyde to an alcohol. And the alcohol that we produce from D-glucose is called D-glucitol. Now, D-glucitol has a common name, sorbitol. Sorbitol is found in cataracts. If we use mannose and make D-mannitol, that's used as a medium in biological materials. And D-glyceraldehyde can be reduced to glycerol, which is a key component in fat and many lipids. Interesting to note is now the molecule somewhat symmetrical. If I took L-gulose and did this reaction, I would get L Gulitol. Now what's interesting about this is L-gulitol and D-glucitol are the same molecule. The symmetrical groups at each end allow you to freely rotate this molecule this way. If you rotate it that way, the D-glucitol becomes the L-gulitol. And so the distinction, because this aldehyde becomes a CH2OH between L-gulitol and D-glucitol, disappears. Now, there are molecules that consist of alcohol groups off every carbon in a cyclohexane ring. And if you have 1, 2, 3, 4, 5, 6 hexahydroxy cyclohexanes, then you have a molecule that's called an inositol. An inositol. Inositol are not true carbohydrates because they lack the oxygen in the ring, but this molecule, called myo-inositol, is an example of a type of molecule that is found inside the cell of almost all living species. It's found in the lipid phosphatidylinositol and phytic acid, and the hexaphosphoric acid ester of inositol. The calcium-magnesium salt of phytic acid is referred to as phytin. And so this is a molecule very similar to what we see in carbohydrates, and as a result is referred to here. Now, another very important derivative of sugars are the phosphoric acid esters. the sugar phosphates. The sugar phosphates. And sugar phosphates, or phosphoric acid esters, are formed by the reaction of sugars with the inorganic phosphate of some high-energy compounds, such as ATP or ADP. And since most monosaccharides are metabolized as phosphate esters, these esters are found in all mammalian cells. Now, using the de Bruijn-van Eckenstein transformation, you recall that was the methodology by which fructose could interconvert from glucose. You can obtain inorganic phosphate esters of both glucose and fructose. For example, if I take D-glucose and I react it with an enzyme phosphate, I'll just put enzyme. There are enzymes that will phosphorylate the glucose molecule at the 6th position. And we would get what is called, if we start with D-glucose, we produce what's called D-glucose 6-phosphate. Now, remember that glucose is in equilibrium in base with the enediol intermediate. Now, except in this case, the enediol intermediate has this phosphate group in the 6th position. And the enediol can interconvert to produce fructose, except this fructose has the phosphate group at carbon 6. Now, if the fructose reacts with a phosphorylating enzyme again, the CH2OH at carbon 1 can react and you can produce D-fructose 1,6-diphosphate. So what we have here is D-fructose 6-phosphate, and what we have here is D-fructose 1,6-diphosphate. Okay. Now, these sugar phosphates exist like monosaccharides in ring form. But note that the term pyranose is not used in the nomenclature, and the formation of the phosphoric acid ester or the phosphate ester is dependent on the pH. So we can actually synthesize these sugars in ring form, and I'm going to draw the Howarths of that. So if I have alpha D-glucopyranose, and it enzymatically reacts with phosphoric acid, so HO is phosphoric acid, H3PO4. I can produce what is called alpha D-glucose 1-phosphoric acid. And that's from alpha D-glucopyranose. Or, depending on pH, if the pH is higher, that is, we're under more basic conditions, the phosphoric acid will be deprotonated and become a phosphate. And this would be called alpha D-glucose one phosphate. Now, sugar phosphates are intermediates in many metabolic processes. The degradation of glycogen to lactic acid in muscle glycolysis, the fermentation of sugars to alcohol, and the formation of carbohydrates by photosynthesis all involve sugar phosphates. Several keto sugars as phosphate derivatives are important intermediates in photosynthesis. Fructose occurs as a 6-phosphate and a 1,6-diphosphate. Ribulose occurs as a 5-phosphate and 1,5-diphosphate. And cetoheptulose occurs as a 7-phosphate and a 1,7-diphosphate. And let's not forget the case of ribose. Ribose occurs in RNA and 2-deoxy deribofuranose, occurs in DNA. And alternating between the ribose fragments are phosphate groups. Sugar phosphates are the key constituents of the backbone in the structure of RNA and DNA. Now, the last biologically important carbohydrate derivative, monosaccharide derivative that we want to look at is the amino sugar. Amino sugars are monosaccharides in which the OH functional group has been replaced by an NH2. For example, consider alpha-D-glucopyranose. So here's alpha-D-glucopyranose. Now remove the OH and replace it with an NH2. Do we refer to this as 2-amino alpha-D-glucopyranose? No, we do not. Here once again is alpha-D-glucopyranose. If we were simply to say 2-amino alpha-D-glucopyranose, there would be no indication that the OH has been removed. You'd be simply stating that there is an amino, a carbon 2. So this would be 2-amino alpha-D-glucopyranose. But if you want to indicate that the OH has been removed and replaced with an NH2, you must state 2-amino 2-deoxy alpha-D-glucopyranose. Actually, let's put this down. Okay, so we'll put a hyphen here. Alpha-D-glucopyranose. Now these amino sugars are important. And by the way, the common name for this is simply glucosamine. Glucosamine is an important precursor to the component found in the exoskeletons of insects and crustaceans. It's a key constituent found in chitin. And we can do this in the laboratory. We can modify the amino group to an amidyl group, N-acetyl, by reaction with acetyl chloride. So if I take CH3, C double bond O, Cl, and react it with this, I can produce N-acetylglucosamine, Now, this amino group is often acylated, such as I've shown here. There are more than 60 amino sugars presently known, many of which have been isolated and identified as components of antibiotic or anti-cancer compounds. You should be able to name and draw amino sugars. The last molecule I want to look at really isn't a biologically important derivative other than the fact that it is useful in the laboratory. Before the advent of spectroscopic methods, there were qualitative tests for the identification of carbohydrates. One of those tests was periodic acid oxidation, and we've talked about that in the last Scribblecast. But another methodology for aiding in the identification of a sugar involves the production of osazones. Now, osazones are products of the reaction of monosaccharide and phenylhydrazine. Hydrazine is two NH2 molecules bound together. This molecule is hydrazine. Now, if I take one of the H's off this molecule and attach a phenyl, I have the molecule phenylhydrazine. If I take phenylhydrazine, and by the way, there's no space here, it should be one word, sorry about that. If I take phenylhydrazine and react it, for example, with D-glucose, so I take D-glucose, and I react D-glucose with phenylhydrazine, two phenylhydrazines, and I react it at 100 degrees C in acid. I will produce a phenylosazone. This molecule would be called D-glucose phenyl Osazone. One word. Now, D-glucose phenyl Osazone is a unique molecule because these phenyl Osazones are very white, colored, bright colored, orange, yellow, red, crystalline materials that can recrystallize really nicely and give you very pure compound for which you can get very accurate melting points. And based on the data, you could carry out a melting point on the phenyl Osazone and very easily narrow down the identity of the unknown carbohydrate. I say narrow down because note that if I had D-mannose or D-fructose, they would produce the same phenyl Osazone. D-glucose, D-fructose, and D-mannose all vary the structure here. And since they only differ in structure in that position, and that's where you produce the C double bond N, those three molecules would all give the same phenyl Osazone. So in other words, D-glucose phenyl Osazone, D-mannose phenyl Osazone, and D-fructose phenyl Osazone are the same molecule. Again, this is only useful in laboratory identification. If you were given an unknown carbohydrate, you would take the melting point of the phenyl Osazone, you would look it up, and this would narrow down the identity. You would then know you are working with either D-glucose, D-fructose, or D-mannose, and you could carry out other tests to further identify. Well, that's it for biologically important monosaccharides. Next Scribblecast lecture, we're going to move on and examine disaccharide chemistry. I'm Dr. Brian Lloyd. This is my Scribblecast. I thank you for listening.